Hello, everyone, and welcome to our virtual field trip. I'm Teresa, one of the board members at Washington's National Park Fund, and I'm thrilled to be your host today. Before we start, there are a few housekeeping items to cover. This field trip is 45 minutes in length with discussion from our speaker and a Q&A at the end. Feel free to enter any questions in the Q&A box as we go, and we'll get to as many as we can at the end of our time together. Closed captioning is enabled, which you can access at the bottom of your Zoom screen. We welcome any feedback as we work to make these trips as accessible as possible. For those new to Washington's National Park Fund, also known as WNPF, we are the official fundraising partner for Mount Rainier, North Cascades, and Olympic National Parks. We have the honor to work closely with park leadership to fund 30 to 40 priority projects each year. And this year, we're excited to announce that we've reached a record-setting year of support with over 2 million awarded to our parks for projects that fall into four core areas: advancing science and research, improving visitors' experience, expanding volunteerism and stewardship, and embracing inclusion. Thank you. Today, we are thrilled to be joined by Allie Stift. Allie is a PhD student at Washington State University in the Wild Herbivore Ecology Lab. Her research broadly focuses on wildlife habitat use, and her PhD dissertation centers on modeling rocky habitat characteristics and assessing habitat suitability for rock-dwelling wildlife in the Cascade Mountains. When Allie began her PhD in 2024, researchers at WSU and Mount Rainier had already started discussing the potential for a Pika-based community science project for a while. However, they needed someone to lead the project, analyze the data, and communicate the results. Allie was a natural fit for the role, as she had not only has a background in environmental education and wildlife research, but she's also incredibly passionate about the subject. The timing aligned perfectly, allowing her to take on the Mount Rainier Pika Community Science pilot season as a session of her dissertation work. When she's not working on her research, Allie can often be found doing more of the same things she's paid to do, hiking and camping in the mountains. Last summer, she spent a week collecting data at Mount Rainier and then headed straight to the enchantments over the weekend. When she's not hiking, camping, or stalking pikas, Allie enjoys traveling and foster cats from the local animal shelter. Allie, we are so excited to share the time with you today to learn more about your important work, the habitats that you study, and of course, the adorable subject of today's talk, the one and only Cascade pikas. Allie, when you're ready, the floor is yours. We're so excited to learn more about your work.
Thank you, Teresa, for that wonderful introduction. And thank you all for being here today. I am incredibly excited to be talking with you about the Mount Rainier National Park Pika Community Science Project. But first, I'm going to go into a little bit of background on our study species, the American pika. This is the American pika. If you've gone on a hike in alpine regions of Washington, there's a good chance you've seen or heard one. These 6 to 8 inch animals can be identified by their brown fur, large Mickey Mouse-like ears, potato-shaped body, and of course, their distinctive high-pitched calls. American pika distribution ranges across 10 states in the western US and into Canada. In Washington, pikas are found throughout the Cascade Mountain range at high elevations, such as in the North Cascades, all the way down to near sea level at the Columbia River Gorge. The habitat that pikas are most commonly associated with is talus, a rocky habitat found in high alpine or mountainous areas. Although the pikas have been recorded using a range of broken rock habitat, including lava flows and even some man-made features as well. Pikas generally prefer to eat grasses and wildflowers in alpine meadows, but they also eat mosses, shrubs, and even pine needles. One of my favorite fun facts about pikas is that they do not hibernate. They store all of their food for the winter in big piles of vegetation under rocks called hay piles. In the U.S., the American pika's conservation status varies from secure to vulnerable to imperiled. According to the International Union for Conservation of Nature, or IUCN, pika populations across their range are stable but decreasing. One of the biggest threats to pika populations is climate change, as pikas are cold-adapted species that are unable to function at high temperatures and depend on snow cover in the winter for insulation and protection. In Washington, pikas are listed as a species of greatest conservation need, but their population size is currently unknown. Long-term monitoring projects are needed to gain a better understanding of pika population sizes and the effects of climate change throughout the state. Mount Rainier National Park is located in west central Washington, around 60 miles southeast of Seattle, and it is home to one of the most iconic pika populations in Washington. While there have been some previous pika monitoring efforts conducted in the park, current population trends are unknown. To inform management and conservation strategies, widespread monitoring is needed to determine whether populations are stable, increasing, or decreasing, as well as where they're located within the park. But this kind of large-scale monitoring requires a lot of work, and Mount Rainier National Park is limited in the number of staff they have to accomplish this large task. As a solution, Mount Rainier National Park is implementing a pika monitoring program in partnership with community scientists, which had its pilot season this past summer. Now, why is having community scientists monitor pika populations at Mount Rainier such a fantastic idea? Well, Mount Rainier gets on average around 2 million visitors every year. Now that is a lot of eyes and ears that could potentially be spotting pikas. Pikas are relatively easy to spot and identify compared to other animals, as they are active during the day, very active on the top of their habitat or talus, and they tend to call or yell at people passing by, which makes it pretty easy to notice that they're there. Additionally, as I preluded to before, these doing community science monitoring in Mount Rainier can lead to a long-term, so over several years, widespread across the entire park data set that could help to inform management and conservation strategies. The goals of our 2025 pilot season was to record American pika occupancy, or whether they're present or absent in an area, abundance, the number of pikas within a site, and habitat characteristics in Mount Rainier, compare volunteer and wildlife staff occupancy and abundance measurements, assess new methods for recording habitat characteristics using cell phone images, and inform the protocol for a full-scale implementation of our American Pika Monitoring Community Science Project for future years. So I am going to go through each one of these goals, talk about the methods that we conducted, and then explain the results. And so it'll go goal, method, results, goal, method, results. And I'll keep referring back to this slide to keep you right along with me as we go through all of these goals. So to start, I will talk about our goal for recording American pika occupancy, abundance, and habitat characteristics. To begin, we recruited volunteers by sending out an interest form that collected information on their previous pika identification skills, whether they had never seen a pike before, did not know what it looked like or heard like, or sounded like, if they could identify pikas by sight, or if they could identify pikas by sight and call. And we collected this information to get at our second goal, which I'll talk about in a little while, but we wanted to know what stage of experience people had coming into this project. We had 140 people fill out the interest form, and we randomly selected 65 to be trained in June and July on alpine mammal species identification skills and safety in the field. Now, Pika's surveys consisted of a volunteer and a wildlife staff member listening and looking for pikas at a talus site for 30 minutes and measuring occupancy, the number, whether there's pika there or not, and abundance, the number of pikas within a site. Now, a wildlife staff member was either myself or my technician, Anna, who served as a reference to compare volunteer data to, because we wanted to know how data collected by volunteers compared to data collected by pika researchers. So the wildlife staff member and a number of volunteers went out to a survey site and recorded location, date, temperature, and weather, the number of pikas seen, heard, and total number of pikas within a site, the time a pika was first heard and seen, any indirect signs of pikas, such as their fecal pellets or hay piles, and then their confidence level. And all of this information let us know the occupancy and abundance within a site, anything that may be interfering with our ability to record occupancy and abundance, as well as some information on how well our protocol might work. To record habitat characteristics at each survey site, we used a new technique that measures rock characteristics from a cell phone image. Now, what this looked like in the field was after the pika survey, volunteers took a picture of the rocks at the survey site that included a reference target in it for scale. And right here is an example of that reference target. The images were then processed using Python software coding called segment everygrain, which identifies each individual object in the picture, for in our case rocks, and creates an outline around each object so that we're able to measure that object. And how we get those measurements is in the code, we outline the points of the reference marker, and we tell the code from this dot right here all the way down to this dot, that is 15 centimeters. And then it is able to measure the reference marker, that 15 centimeters, and put it in reference to the size of the pixels in the image and produce an Excel sheet of measurements for each individual rock that we have outlined here, with a measurement that ideally is incredibly similar to the measurement that we would collect if we were measuring out in the field. From these rock images, the 4 measurements that we're able to collect is perimeter, area, longest axis, and shortest axis. And this habitat data can be useful to understand the relationships between the type of rocky habitat and pika occupancy and abundance. For example, previous research has shown that pikas select for specific sized rocks for creating their hay piles and establishing their territories. So what this looked like in the field is after the volunteers completed the pika occupancy and abundance surveys, we had them place the reference marker in the rocks, take a picture of the rocks from 3 feet away, from 9 feet away, and then collect an image of the entire site. And this gave us some up-close images of the rocks, some pictures from a little bit farther away, and then one of the entire site to really give multiple dimensions of exactly what we're looking at in these images. And moving on right into our results for this first goal, we had 51 volunteers complete between 1 and 6 surveys across 20 survey sites. And this led to 114 volunteer data entries, which after cleaning led to 96 data entries for analysis. And then we had 60 staff data entries. We detected pikas at 19 out of our 20 survey sites, and abundance recorded at a given site ranged from 0 to 15. And we collected over 350 habitat images. So this first goal was definitely achieved. We collected a lot of data this past summer. And then, so for this second goal, we took a little look into the data that we collected and compared volunteer and wildlife staff occupancy and abundance measurements. Now, as I mentioned, when we had volunteers fill out the interest form, we had them state whether they had no previous experience identifying pikas, whether they did not know what a pika was, whether they could identify a pika by sight, or whether they could identify a pika by sight and call. And what we really wanted to determine was, how does the data collected by volunteers of previous experience levels differ from wildlife staff? Does it matter if someone has completed 100 surveys or 2 surveys? Can they count pikas at the same accuracy level? And ideally, yes, because everyone took our training, and if it worked, it put everyone on the same base skill level to be able to record pikas. Thus, this was really a test for how well our training worked. And how we compared this data is we compared occupancy and abundance measurements from any given survey between the volunteer and wildlife staff member that was there, and then we also compared measurements across the 3 volunteer ID skill levels. Now, to compare occupancy measurements between volunteer and wildlife staff, we calculated how many surveys volunteer and staff data matched. Now, data can match between volunteers and staff for 2 reasons. There is a pika there, and both the staff and volunteer correctly record there's a pika there, or there's no pika there, and both the staff and volunteer say, yep, no pika. Alternatively, data cannot match if there is a pika there or not there, but the wildlife staff and volunteer data differ. To compare between volunteer and wildlife staff abundance or pika counts within a site, we calculated the difference by subtracting the wildlife staff count from the volunteer count. Now, what this looked like was, if a volunteer counted more pikas than wildlife staff, say 8, compared to 6, the difference was positive, or positive 2. If they counted the same amount of pikas, say 3 and 3, the difference was 0. If the volunteer counted less pikas than the staff, say 0 and 1, the difference was negative, or negative 1, and so on and so forth. And we calculated this for every single survey. And our goals for both the occupancy and abundance were that they were as similar as possible. So we were hoping that as many surveys matched for occupancy, and then for the difference, we were looking for very small numbers, ideally as many zeros as possible, because that would mean everyone at the same survey was counting the same amount of pikas. And then to compare across ID levels, we created an Excel spreadsheet that had every surveyor and then their ID skill level, whether none, sight, or sight and sound, whether their occupancy classification on that given survey either matched or did not match, and then the abundance difference. And we ran statistical models to see if there were significant trends with one group or another group of ID level that were with the abundance and occupancy data. For example, if the ID skill level of people who had never ID'd before were consistently measuring less pikas than the wildlife staff, that would indicate to us that something funky is going on. So we wanted to look into that. And right into our results. Our occupancy estimates matched for 90% of the surveys, so 86 out of the 96 surveys, which is fantastic. And so we had 10 surveys that did not match, and this was exactly what we were hoping to see. Abundance estimates were similar between volunteers and wildlife staff, and that can be seen right here with the majority of our surveys not differing and having a count difference of 0. And the ones that did differ typically differed by around 1 or 2 pikas. Once again, this is fantastic, exactly what we wanted to see. Looking between the survey ID or the previous ID skill levels, for occupancy, we did not really see a trend in any of the 10 surveys that did not match for any of the specific IDs. As you can see, they're pretty evenly spaced out, and so that lets us know that ID is most likely, previous ID skill is most likely not influencing occupancy measurements. And then for abundance measurements, we compared mean difference, which is on our y-axis over here, and to ID skill level, which was on our x-axis. And what we were really looking for is that mean difference across these three groups were as similar as possible. And this is exactly what we saw. As you can see right here, they're very similar, very close to 0, which is our wildlife staff measurement. And the significant error bars overlap, indicating non-significant difference. Which that is great. It means everyone is surveying at the same skill level and at a pretty high one as well. So that is awesome. All right, so as I've said many slides for your past results, this goal was definitely a success. Moving on to our third goal of assessing new methods for recording habitat characteristics using cell phone images. As a reminder, we collected habitat characteristics using a segment by having volunteers take pictures of the rocks and running those pictures through segment evergreen, where we're able to calculate 4 measurements: perimeter, area, longest axis, and shortest axis. But these methods have never been used before in a community science setting, and so we wanted to know, does the measurements that we're getting differ depending on who is taking the picture? Ideally, they're as similar as possible. That way, no matter who's going out to record this data, we're getting the same results. But it could vary from person to person, and we wanted to look into that. And how we did that was we had volunteers, after their survey, collect a picture of the rocks in the same location from the same distance of the same rocks. And so, for example, these are 3 different volunteers, surveyor 1, 2, and 3. They were at the same spot on the same day and collected this image of the rocks from 3 feet away. But as you can see, they do kind of differ in the angle that they were taken, and so we wanted to investigate what the effects that might be. And then to give another example, here are 2 pictures taken by 2 different volunteers, but these ones almost look identical. They're very similar. And then for a third and final example, 2 images taken on the same day of the same rocks, but by 2 different people. And how we analyzed it is we ran the images through segment evergreen and extracted the measurements, and then compared the measurements of the same rock but from the 2 different images. So for example, we compared the measurements of this rock and this rock, same rock, 2 different images, between this rock and this rock, and this rock and this rock, and so on and so forth. And how we did this is each rock had an individual label. And so, for example, this is rock number 9 and this is rock number 16. And in our Excel spreadsheets that get output from segment evergreen, we have each individual rock with its 4 measurements: area, longest axis, shortest axis, and perimeter. And so we lined up each rock with its similar rock in the second image by the label. And so back over here, we've got rock 9 and we've got rock 16. And then right here, we've got rock 9 with all of its measurements and rock 16 with all of its measurements. And so each of these lines are the same rock, but just from different images. And we calculated the percent difference between each measurement for each pairing of rocks. And this is what it looked like. So we've got rock 9 and rock 116. The percent area, or the area between the 2 images, differed by around 9%. Longest axis differed by around 2%, and so on and so forth. And so we did this for every pairing of rocks that we could connect in every group of images. And so this is one group of images, and then we averaged to get an understanding of across the 2 images, how much are these varying across all of the rocks. And then we did this for 10 different 10 different pairings of pictures, so 10 different surveys. And then we average across those to get overall, right off the bat, what are we looking at? How much does the percent difference between rocks really differ? And here are our results. Across 10 survey sites with paired images, our percent difference between rocks ranged from around 25% in area down to around 13%. And while these numbers are good, they're a little higher than we were hoping to see, which was around below 10% error. Now, this larger error is likely due to because the methods that volunteers are collecting the images is differing, most likely an angle, and so volunteers are tilting a little up or a little down, despite the protocol. And so what this indicates to us is that we need to modify our protocols and training so that in future years, directions are clear on how the photos should be taken. So they're taken in a much more uniform way. And we already have plans to implement this tweaking of the protocol and training for this upcoming summer. And so this assessing our new methods was still a success. We learned how well these methods work, and we gained some information on how to improve it for this upcoming summer. And speaking of this upcoming summer, that kind of leads me to our fourth and final goal of informing the protocol for a full-scale implementation of the project. And in order to kind of review this goal, I'm going to go over what we learned and how this is going to influence our protocol for this upcoming summer. So for our first goal, what did we learn? We learned that volunteers can accurately measure pika occupancy and abundance. And this is very exciting, and so I have a party pika to represent it. For our second goal, we learned that data does not vary due to previous experience level. And then for our third goal, we learned that rock habitat can be measured, but refinements to the protocol is needed. Now, all of these three things that we learned from this previous goal is going to inform our protocol, and it already has, for this upcoming summer. Now, what this upcoming summer will look like is we will be continuing our Pika Community Science Project, and we are recruiting volunteers for our VIP program. And the VIP program is Volunteers in Parks, and anyone who's interested will complete a 2-hour training, or people who are chosen will complete a 2-hour training in late June and early July for in-person, and Zoom options will be available, and they'll commit to at least 2 days of pika habitat surveys between July and October. So people who sign up and are selected for this program will be full volunteers at Mount Rainier National Park. And the volunteer requirements are that the volunteers are 18 and plus, have the ability to safely navigate to and from survey sites, represent the National Park Service in a professional manner, and follow the park's policies and guidelines. Now, I'll have an interest form QR code on the last slide of this presentation that also has more details on the project as a whole and what it entails, and what's expected of volunteers. It closes on May 25th, and selected volunteers will be notified by May 30th. And anyone can fill out this form as long as they meet the requirements. But I need to stress that we do have high levels of interest, and unfortunately we are limited in the amount of volunteers that we're able to accommodate just due to the amount of supplies and resources the park has. And so we will be completing a lottery system to select new volunteers, but unfortunately we cannot guarantee anyone a volunteer position. Volunteers who are not selected will also be notified by May 30th. And so we also are launching a new program this upcoming summer for recording pika locations within Mount Rainier National Park that anyone can do for anyone who's coming to the park using the Pika Patrol app. The Pika Patrol app is available and free for anyone to download. It's a simple form for documenting pika locations and habitat, and it automatically records location, date, and time, and you can upload pictures and videos. And so it's a great way to be able to record locations of pikas really anywhere in Washington. But we've partnered with the creators of Pika Patrol so that we can receive the data of the pika recordings within the park. And so we are advertising this as a fantastic way for anyone coming to Mount Rainier National Park this summer to download the app and record pikas. And we will have 15 to 30-minute virtual trainings on how to use the app and what we're looking for, specifically at Mount Rainier in July. And be on the lookout because we will be advertising those trainings soon. And the Washington National Park Fund has agreed to also advertise them. So however you heard about this talk, take a look there. Because we will be spreading the word. And so this is a option for if you are not selected for the VIP program, but still want to be involved and are going to to Mount Rainier this summer, or if you're not looking for a large commitment for being an official volunteer, but you're coming to Mount Rainier and you want to record pikas. So as my final point, I will just reiterate that all of this data collected by both our volunteers and through the Pika Patrol app will help us gain a better understanding of pika populations within the park. Are they decreasing, increasing, or stable? If they're decreasing, why might they be decreasing? Because pikas are climate-sensitive species, monitoring populations allows us to track potential population declines due to climate change or the threat climate change may pose in the future as well. What habitats are they using throughout the park and where are they located? All of this information will lead to informed, effective management and conservation of American pikas within the park, which at the end of the day is our main goal. And so with that, I would like to say a huge thank you to our 2025 volunteers and staff, the Washington's National Park Fund, Cascade Pika Watch, the National Park Service, and Washington State University for all of their support throughout our first pilot season and into the future. And with that, I will take any questions. We've got our QR code for the 2026 Mount Rainier Pika Project VIP interest form on the left and a QR code to download the Pika Patrol app on the right.
Thank you so much, Allie. That was amazing. I am so excited. I was already excited for summer, but so much more excited now. And many of the places shown are familiar as a lover of Mount Rainier National Park, and I'm sure that is the same for many members of our audience. It's such an amazing place. Excellent. All right, we've got a few questions from our attendees to talk through. We'll start with a question around your process for selecting specific sites to survey. Why or why not do you choose various sites in the park? Can you talk more about the methodology?
Yeah, so for this past summer, we wanted to survey in areas where we knew there was potential for pikas. And so we got a GPS location data form from Mount Rainier that had over 900 potential pika locations within the park that had been documented over the past, I think, 30 or 50 years of different habitat or different areas where it had proper habitat that pikas could be, or pikas were seen there at some point, but we don't know if they're still there. So I used that as a starting point. And then I filtered it by distance to trail, because for this project, for our volunteers, we stayed on the trail, and then distance from the visitor center, because unfortunately we do not have the time or the capacity to be hiking super duper far, at least this past summer. And so I narrowed it down through that and then took a random selection spaced out by the different watersheds throughout Mount Rainier, which I believe there's around 7 different watershed boundaries. And so that way our data points were spread out across the entire park, but still randomly selected.
Wonderful. Thank you so much. And follow-on question to sampling on those sites. We have a question asking if you're planning to do the same type of sampling this coming summer as was done last year. For instance, are we still planning on doing 30 minutes in the early morning on given survey days, or is anything changing for this year?
Yeah. So last summer we surveyed pretty early. We hit the trail around 7 a.m. and we surveyed for around 30 minutes. And we'll most likely still be surveying in the morning because that's when pikas are most active. It's the coolest, especially at the highest temperatures of the summer. So we'll be surveying in the morning, but the time that we're surveying might be a little shorter. I still need to process the data, but we recorded, as I mentioned, the time that a pika was first seen and heard. And so if the trends are showing that we were detecting pikas within 15 minutes, then there's no real reason to sit there for a full 30 minutes. But my guess is it'll range around 15 to 30 minutes this upcoming summer.
Awesome. Thank you. All right, got a great question from a pika lover further afield around the pikas in the North Cascades National Park. The question is around whether or not the Pika Patrol app could or should be used for community science sightings outside of Mount Rainier.
Yeah, definitely. Great question. Pika Patrol can be used by anyone, anywhere. And so even if you're in Colorado, in Rocky Mountain National Park, or if you're in Snoqualmie and you see a pika, you can record it anywhere at any time, which is fantastic. We won't necessarily get the data, but I encourage anyone who's a pika lover to download the app because it is truly a fantastic app.
Thank you. I know I will be doing that
Yeah.
this summer. Very excited about it. All right, we've got a wildlife taxonomy question around pikas and some of our other charismatic fauna, our friends the marmots. The question is whether or not they are related at all.
Ah, such a great question. And actually, I had a little tidbit about this, but due to time, I took it out. Well, marmots are rodents, members of the order Rodentia, and it's pretty common for people to make the mistake that pikas are rodents because they kind of look like guinea pigs or mice or like a chipmunk. But they are actually a member of the order Lagomorphia, and so they are lagomorphs, not rodents. And this other members of the lagomorph order is rabbits and hares. So they're actually a little more closely related to rabbits and hares than they are to marmots.
Excellent. Thank you. Love that and appreciate that information. All right. A question on observing them from a general community perspective. You mentioned that early mornings are the choice for your surveys. To follow on that, have there been any patterns on what other than time of day? When is a good time to see them? Weather, temperature, etc.
Yeah, and so they're typically most active when it's coolest out. And so on really hot days in the middle of August at noon is going to possibly be less likely to see a pika. And so temperature plays a huge role into it, and thus time of day, as I mentioned. They do tend to become more active later in the year, so around like September, because they start actively haying a lot. So creating those hay piles, it is go time to make sure they have enough food to make it through the winter. And so you'll see that activity quite a lot of them running back and forth from the center of the talus or from their hay pile to the edge to collect vegetation and then run back. And so that activity level makes them pretty easy to spot compared to earlier in the year.
Wonderful. Thank you. All right, we've got a couple of questions on your volunteer program. Side note, I love that you have more folks than spots, but a wonderful problem. For other interested volunteers, I have a question on how groups are organized for survey days. In other words, where are folks assigned days? Do they choose their own? And how are the days in the field organized from logistical perspective? And secondarily, in a follow-on, is there any identifying information, hats, t-shirts, vests, etc., to indicate that you're a participant in the project when you're in the field?
Yeah, great questions. And so for determining survey days and groups, we put out a spreadsheet where it had all of the information of all of the different sites on distance and side of the park and like hiking difficulty so that people were very aware of how difficult it could be to get to a certain site, or we had some sites right along the side of the road. So people were given information about what about the sites, and then we assigned a survey site for each day. And so volunteers don't necessarily get the choice of exactly which day and which survey site for a specific day, but they can go in and look at all of the dates and look at what survey sites are associated with that date and sign up. And so for grouping, I'm expecting this upcoming summer to have groups anywhere from around like 3 to 6. And so that many are like that amount of people can sign up. And so I'll have kind of a block on an Excel sheet with each date and then each survey site and then a block of up to 6 people. I inform everyone when I'm going to open it up. I open it up and then people can sign up until that like 6-person block. And then sometimes we had people drop out too, and then other people can fill in. And then, I'm sorry, what was the second question?
Is there any identifying?
Oh, yes. Yeah, and so we will most likely not have any identifying gear. When we're surveying close to the road, we wore orange vests, but nothing necessarily to say like Pika people, although that would be pretty cool.
It would be pretty cool. All right, thank you for that information. One more question to bring it home, a fun one. Can you tell us about a memorable experience that you had out in the field, either serving in general or with any special individuals that you were happy to meet that day?
Yeah, definitely. I mean, a couple come to mind. There was this, when this picture was collected right here, where a pike was just actively haying. And so the entire survey was just going back and forth, just carrying these big mouthfuls of food. And we could see it building its hay pile. There was one site that felt like it had like a million pikas in it, which it definitely did not. But all of a sudden, they all started calling at once, and it just made it clear how many pikas are within that site. And then my last one is when we were conducting a survey, we had some people walk by and ask what we were surveying. And one of my volunteers did a fantastic job of explaining the project and what pikas are and why it's important of what we're doing. And it just was such an awesome moment to see the community building and people informing other people about pikas and kind of the future of this project. And so those were my favorite moments from this past summer.
Awesome. Thank you very much. I love those stories. Yes, that's incredible. All right. Thank you so much again. We are out of time for our formal presentation. For those who want to revisit it, you'll have the opportunity to do so. And Allie, thank you again for sharing your time, your expertise, and your passion for these incredible animals and this very important project with us today. So very grateful. Before the day ends, I also want to mention that you can learn more about the projects we fund, the Washington National Park Fund, at WNPF.org. And for participants Thank you so much for joining us for another incredible virtual field trip. If you'd like to see other episodes of our virtual field trips, you can find us on YouTube by searching Washington's National Park Fund, including this field trip to be posted soon. And if you're interested in joining us for future events, whether it's online or in person, you're always invited to visit us at wnpf.org/events. And as always, thank you to our presenter and our participants and our supporters for supporting our parks and the incredible creatures that live within them. Can't wait to see you next time and in the parks.